Hi friends, today I'll be talking about design of combinatorial library. So little I'll be discussing about why do we need this type of chemical libraries, and even we have explored many chemical libraries in terms of drug bank, PubChem and zinc database. We have seen over there this is these are the platforms where we are finding the huge number of chemical compounds which also have that desired properties to become a drugs. And this type of chemical library also have all kinds of drug informations which has been taken from different type of studies, different experimental methods, different in silico methods, different in vitro studies also. As you know that when we explored that chemical libraries in PubChem and drug bank, so you have seen over there what types of data are there, like their drug properties in terms of their physical properties, chemical properties, structure properties, even in structure properties, 2D structure properties, 3D structure properties, and so on. And that synthesis methods and the mechanism of drug actions into the target site, their target informations, their functional group informations, and many more which we have explored before, right? And their clinical trials datas will be there, their nomenclatures will be there, right? So many more informations which is been stored in one platforms is called the chemical libraries. So the objective of this type for making this type of library is to identify sets of compounds, a list of compounds which we can say the potential compounds, right? And make a combinatorial libraries for these compounds which also should respond some drug likeness properties. So what is that combinatorial libraries? So it defines as a chemical compounds which can be ensembles as a library, and screening of those libraries for the compounds which also must have that desirable properties to become a drug. So before coming to this wide discussions, we need to understand that chemical space. Like what type of informations can be stored in a one libraries, in a one database, in a one common junctions, which can be accessed by the people who are in these fields, by clinicians, by authors, by scientists, by pharmaceutical industries, and the, many others databases, uh, authorized peoples who can explore and access and retrieve the data with the cross references. So what is the chemical space? Chemical space is that organic compound, a small molecule in organic chemistry, a small molecule in organic chemistry. So it's of course the small compounds we should be like organic compounds. So we need to understand those compound, and we need to understand the chemical space in terms of that their chemical synthesis, drug design methodology, drug design approach, and of course we have discussed many approach, of course, in terms of in silico analysis, right? So like a docking studies, QSR studies, right? Ligand-based drug design approaches, structure-based drug design approaches. These all these things which we have discussed to identify the lead molecules, right? Potential molecules which have that ability to become a drug. So, and that how these compounds also will be responding for a particular genes and for particular entire genomes, and their linkings, either these compounds will be targeting to a particular genes, right, interacting to a particular gene. So we need to understand that entire mechanism of actions, okay, how molecules, those molecules work inside the cell and tissues, their interaction mechanism, and that is called the system biology, right? Nanotechnology. So how these drugs and these compounds can be used at the nano levels, so that it will be useful even incorporating these drugs at nano levels for treating for a particular disease and any other. So first we need to understand that why do we need this type of chemical library design. So already we know that even we have this experience to generate huge and enormous datas, right? So maintaining the datas, right, and the planning of the datas, right, monitoring of these datas, right, and any kind of performance about those datas, right, and the reporting of these datas is are important, right, and even the validations of the data sets are important, that should be accepted by the pharmaceutical experts, right? And we should also have that enough knowledge, okay, how we are going to transfer our data into informations, and that informations can be passed to the knowledge, and that knowledge can be implemented for a particular purpose or useful purpose. So that things we need to understand that. So data are there in that, in terms of the chemical libraries, how we will be exploring the data, how we'll be retrieving those data, how we'll be converting all the information into knowledge. That things also should be there, and the knowledge can be used for a particular useful purpose. So now we need to also understand about the relationship between that structural properties and their biological response, right? Their mechanism of actions, their reaction conditions, right? So all these things we need to understand when we will be having all type of informations in one platform, right? So what are the scopes are there for making this combinatorial library design, in terms of the representations of the structures, how to search their structures. It may be that similar kind of structure searchings will be there of those compounds, of substructure search will be there, or you are looking for identical structure searching methods. So many more methods, fragment based search is there, so which you are looking for, that okay, that even the substructures are responding well for a particular target. So those type of information if we need, so definitely we need to have one platforms where we can gather or retrieve all kind of informations, right? So these are the methods like similarity search in clusterings. Clusterings also, we need to see the set of compounds which are responding in a similar way, right, in one clusters, and the diversity variations which are responding differently. So those type of things will be there, and searching the databases, computer-aided structure elucidations in terms of their representations, their nomenclatures, their identifications, right? Their international chemical identifiers, right? Their smile notations. So all type of things we can also can get it from the databases. Even the 3D substructure searchings through the pharmacophore modelings and QSR modelings, 3D QSR modeling to find, identify the similar type of compounds which are responding in the same way, right? And QSR and dockings, which we have studied before, right? Interactions of two molecules, and QSR in terms of that is a mathematical functions which correlate wide variety of structural properties with the biological activities. So those type of informations, if we get in one platform, that will be useful for that researchers and the pharmaceutical and people. So kinds of chemical libraries, already we have explored the drug banks, PubChem, and the zinc databases, and many more, right? For the small molecules, even the big molecules, we have explored protein data bank and nucleic acid data bank and many more. So similar type of databases are available also, like literature databases, patent databases, right? Some small project, small molecule specific databases are there, public databases are there, and some proprietary and project specific data are also will be there. So these data and their resources, if you can explore, that will be very useful for you to understand that what are those informations can be explored in a meaningful way. So here we see that like chemical based data mining and explorations. So here also how the chemical of interest, we say that chemical of concerns are responding their activities in between. Many phases are there in terms of that. If you need some suitable compounds from the databases, we need to do that, explore the databases and do some mining for exploring informations. And then if we need like in for chemical specific data, that also we need to explore and mine the datas. And then we need to see that structure also in terms of their structure analogs, property analogs, and how those analogs and the structure are responding towards that biological functions and their mechanism of interactions, which will be establishing that structure activity relationships. So those all these things we can retrieve from that databases. So what are the applications are there for this type of databases, for this chemical libraries or combinatorial libraries? First is that chemical informations. So we need to retrieve that chemical informations, where we can store, retrieve the chemical structures and associate data to manage that floods of the data. And then we need to advertise those type of informations at that in web information, web protocol, or we can say on the internet, in terms of the disseminations of the data on the internet and cross linking of the data informations throughout the databases. Even we can like link all type of information with different type of databases, where any person can explore all informations by exploring even one databases. So like if suppose drug bank information can be explored by using the PubChem, and PubChem informations can be explored by using the drug bank, and even the PubChem and drug bank information can be explored by using the zinc databases. That is also that is called that cross linking of the data to information. And all field of chemistry is right, where we are talking about their physical, chemical and biological properties of the compound, and even we can predict that type of properties of any chemical compounds by giving giving as an input either that nomenclature name or smile notations, or maybe international chemical identifier in terms of the enzyme, or anything where we are predicting their molecular weight. First screening method is the Lipinski rule of fives, where we are screening the compound based on this, like their molecular weight, like log p value, number of hydrogen bond acceptor atoms, number of hydrogen bond donors, and log p values and number of rotatable bonds, right? And their inhibitory properties, right? Enzyme inhibitory properties, and any IC50s, all right, any LD50s, or many more informations we can get at that first screening level. So now another terms in terms of the bioactive molecules, where we can see that that drug interactions into the binding site or target site, right, where drugs are responding well for a particular disease, right? So identifications of new lead structures, right, by different methods which we have studied before, right? And once we have found the lead compound, the next step is that optimizing of those lead structures by different protocols which we have studied before, right? And then make that good structure activity relationship between that between those predictions and that observed biological response. So these are that structure activity relationship where we are mapping like how that structural properties are responding to that biological activities. And then next step is to like comparisons of the chemical libraries, how the chemical libraries are accurate, right? And validate, that also is there. So all the chemical libraries should go through that quality check and quality assurance and monitored by their organizations always, right? And then and also we can see that definitions and analysis of structural diversities, how the structures are responding in similar fashions, or some variations are there. And then planning, so all these things like in terms of that designing, managing, organizing, reporting, and all these things, of course we need to take care for designing that chemical libraries. So now analysis of high throughput data, like high throughput data, how to analyze which are having highly significant statistical values, right, where we have screened thousands of compounds together, right? So those things also can be like taken into considerations while retrieving and exploring the data, right? And docking of a ligand into receptors, those type of data will be there. Predictions of metabolism of genobiotics, right? So all the metabolism, right, and catabolism, right? Metabolism, anabolism, and catabolism already you know. So and our body also have some kind of xenobiotics, right? Some kind of like antibodies are there, antigens are there, enzymes are there, right? Different type of enzymes responding well for a particular purpose. Even type of food additives are can be considered as a xenobiotics, and some type of drugs also can be considered as a xenobiotic. So those type of things we need to understand. And then analysis of biochemical pathways, if the drugs are responding for any chemical pathways or biochemical pathways, we need to understand, and these all type of informations will be there in the databases. That's why we need that chemical libraries or combinatorial libraries. um, uh, and last step is that organic chemistries, where we are talking about that predictions of the courses and the products of organic reactions, right? Like either those compounds are drug are responding reversibly or irreversibly, right? Their mechanism of actions we need to understand, and the design of organic synthesis, right. So how this molecule has come in origins, in existence, by which methods, those things also we need to understand. Of course, all the chemicals are resourced from different type of plants, animals, fungi, and the bacteria, virus, right. So we need to understand that their like experimental studies, right, and that chemical synthesis, like which method it is synthesized by. Many experimental methods are there, many computational methods are there to synthesize, right. Already we have studies all this, how to identify the lead molecule, how to validate, optimize the lead molecule, how to identify the target, how to validate the target. These all these things we have discussed before, well, right. So now here in that organic chemistry, if you say that the predictions of the course and product of the organic reactions, if we talk about, so this mechanism of reactions in terms of the reversible and irreversible. So many drugs which are like binding reversibly and irreversibly. So these things we need to understand. Now all the researchers are trying to synthesize the molecules which have that potential properties to become a drugs, which they are screening by reversible reactions, where reversible reactions have many benefits in terms of that the reversible mechanism. If you say that it's going to bind to a particular enzymes, right, or particular targets, it is not going to bind for a long time. It will give the effect and it's coming out from the target sites, right, so that it's not going to stay long for on tissues and cells, and which will give the less side effect and toxicities, right. While other hands, irreversible reactions which will bind to the target and that any binding sites, and which will of course will be binding tightly, and it will remain stay long for a lifetimes, which will leads toxicities and some side effects. So we need to have to identify such compounds which will be reacting reversibly. So this all these things we need to understand their mechanism. I hope, yeah. Then next also is there the analytical chemistry in terms of that how to analyze their inorganic properties, right, and predictions, their quality, origins, right, all these things and their structures based on the spectroscopic data, and the cheminformatics approaches are there, where we are understanding how to utilize data and how to transfer into informations, and the information can be transferred into knowledge, and knowledge can be passed for a particular useful purpose, right. So I hope you can understand entire about that, the need of that combinatorial library or chemical libraries,